Buenos días. Vamos a empezar ya este acto, este memorial Lino Torre del año 2019. En primer lugar, darles las gracias a todos ustedes por haber venido esta mañana y por haber dedicado una parte de su tiempo para estar con nosotros y acompañarnos en este memorial. Para mí es un especial honor presentarlo este año, después de que durante 16 años lo haya hecho el doctor Luis Grande como jefe del servicio de cirugía. Este es el programa, esta es la estructura del programa que ya conocen. Bueno, finalmente el doctor Julio Pascual ha excusado su presencia, yo haré el cierre del programa y consta, como también saben, la estructura es la misma, es idéntica a la de otros años, con dos elementos principales. Por una parte, una presentación por un ponente invitado, que es el doctor James Kinross, que a continuación les presentaré, y por otra parte, en segundo lugar, la entrega del Premio Lino Torre, que este año es el 20º Premio Lino Torre al mejor trabajo de investigación publicado por un equipo de cirugía de España durante el año anterior, durante el año 2018. Entre medio, en los últimos 3 años hemos introducido un tercer elemento, que es la entrega del premio al mejor residente de cirugía del curso académico inmediatamente anterior, en este caso del curso académico 2018-2019. Pero antes de darle la palabra al doctor Kinross, debemos hacer honor al nombre de este acto y al propósito del mismo, que es hacer un recuerdo, recordar la figura del doctor Lino Torre. Y pienso que eso es especialmente importante. Se van incorporando nuevos cirujanos, y no solo nuevos cirujanos, nuevos profesionales al servicio de cirugía y a otros servicios del hospital. Muchos vienen también de otros cirujanos jóvenes, pero también otros se incorporan de otros hospitales de nuestro entorno. Y realmente pues muchos puede que apenas conozcan la figura del doctor Lino Torre, por lo que creo que es importante que la recordemos, que recordemos la figura de quien fue jefe del servicio de cirugía durante 20 años. El doctor Lino Torre nació en Santiago de Compostela en el año 1923 y vino pronto, con menos de 20 años, vino con su familia a Barcelona, donde estudió en la Facultad de Medicina. Su formación quirúrgica como cirujano la realizó en el Hospital de la Santa Creu y de Sant Pau. Ya pronto, joven, ya destacó, entre otras figuras de la época. Esta nota de prensa aparecía en La Vanguardia, que podría ser un equivalente a nuestros tweets actuales. Ya destacaba la figura del doctor Lino Torre, lo consideraba como una personalidad científica, y junto a otros cirujanos e internistas de prestigio, ya de reconocido prestigio entonces, como el doctor Piulax o el doctor Pedro y Pons. En el año 1966 se convirtió en el jefe del servicio de cirugía del hospital, entonces Hospital de Nuestra Señora del Mar, durante 20 años. Durante este tiempo, ya desde el principio, el doctor Lino Torre demostró una gran habilidad quirúrgica, especialmente en la patología de la vía biliar y también en la cirugía de mama y del cáncer de mama, estableciendo las bases de lo que ha sido posteriormente, con el paso de los años, una unidad de patología mamaria, en este caso conjuntamente con el Servicio de Ginecología, que es un modelo para muchas otras instituciones en Cataluña y también en España. Pero además de una actividad quirúrgica en la cual destacaba por su habilidad técnica, como he dicho, también el doctor Lino Torre demostró un especial interés, además de por la investigación, especial interés por la docencia. Docencia pregraduada, estableciendo los pilares de lo que posteriormente ha sido la unidad docente, primero del IMAS y ahora del PAR de Salud Mar, y por la docencia posgraduada, también sentando las bases de un programa de formación de residentes que ha sido modélico, que fue modélico en su momento también imitado por otros hospitales. Pero sin duda alguna, lo que debemos destacar del doctor Lino Torre fue la capacidad que tuvo para organizar lo que era simplemente un grupo de cirujanos en una estructura organizada, en un servicio de cirugía, tal y como lo imaginamos que es ahora, pero que no existían, prácticamente existían muy pocos entonces. El doctor Lino Torre fue muy querido por todos los miembros del servicio y, de hecho, durante los sucesivos años que iban pasando, pues se celebraban los sucesivos aniversarios. A la izquierda podemos ver el 15º aniversario, a la derecha el 20º aniversario del servicio de cirugía, que además coincidían con el aniversario de su jefatura de servicio. Como reconocimiento a toda su carrera profesional, en el año 1993 recibió el Premio Virgili de la Societat Catalana de Cirugía. Por todos estos motivos, el doctor Lino Torre inicialmente ya estableció las bases de un servicio de cirugía del Hospital del Mar, que posteriormente, con el trabajo de los que le sucedieron en la dirección del servicio, del doctor Siche Serra, del doctor Sánchez Ortega, del doctor Luis Grande, en los últimos 16 años han conseguido un servicio de cirugía con una profunda vocación académica. Y una vez hecho este recuerdo del doctor Lino Torre, paso a presentarles a nuestro invitado de este año. Nuestro invitado es es Mr. James Kinross, quien es Consultant Colorectal Surgeon y Clinical Senior Lecturer en el Imperial College de Londres, que es una institución académica, profundamente académica también. Se formó en el St. Mary's Hospitals y realizó un fellowship de cirugía laparoscópica mínimamente invasiva. Realizó su PhD también en Imperial College de Londres, fundamentalmente en el estudio de la influencia del microbioma en la respuesta inflamatoria después de la cirugía, todo ello con financiación del Royal College of Surgeons y también de la Academy of Medical Sciences. Y también es visiting professor del Royal College of Surgeons de Irlanda. El doctor Kinross, desde el punto de vista asistencial, su interés es fundamentalmente la cirugía del cáncer colorectal mínimamente invasiva y la cirugía robótica. James Kinross is a good example of what a scientific surgeon should be. Besides a very busy clinical practice, he also has shown over the years a very strong interest in research. His research is mainly focused on the study of the gut microbiome, on cancer, and also on surgical outcomes. This is something that we'll have the opportunity to hear today. He also has shown interest on the use of digital tools and also gamification in surgery. As an academic surgeon, he has published a significant number of papers, more than 70 papers in peer-reviewed journals, and also, like this is just a couple of examples, and he has been a recipient of several very competitive grants, like, for example, grants from the National Institute for Health Research, from the NHS, and also from the European Commission, the Horizon 2020 program. We decided to invite James Kinross several months ago based on his academic achievements. I had the opportunity to meet him a few weeks ago in a meeting room, and I have to say that I could confirm that we had made the right choice. So, James, thank you very much for accepting our invitation to be here today and to be our guest for the 21st Memorial Lino Torre Lecture.
So, I have to turn my microphone on. This is the most important thing. And then I will step over my computer. I did not recognize the man in those pictures. I have a lot more grey hair than that man. So we will turn this on, and hopefully this will work. So, ladies and gentlemen, it's my very great honor to be with you at your spectacularly beautiful hospital in Barcelona. London seems like a very long way away. I'm here today to try to explain to you why I believe the gut microbiome is an important new frontier in the way we manage and look after patients. I've shown you a picture of space. Why? Well, because this is actually the whale galaxy. It's many thousands of light years away. It's unimaginably vast and it's ancient, like the gut microbiome. There are more bacteria in your gut than there are stars in this galaxy. In fact, in all of the galaxies, there's hundreds of trillions. They're all connected, they're all ancient. Bacteria were the first life forms on this planet, and they will be the last. You have co-evolved with them since the dawn of time, and they are vital for your human health. And it is genuinely a new frontier. Before I talk, I must just pay homage to Dr. Torre. So, I did a bit of reading of Dr. Torre, and I was not able to understand all of Miguel's introduction, but this picture said many things to me. Clearly, this was a man who valued relationships with people and teams, and this picture says to me that he was well loved. I read his obituary, and it says something interesting in his obituary, that he was always open to all innovation. In fact, he said, even if it meant he would be personally harmed, he would engage with innovation. And he never said no to a new project or idea. And I hope the talk I'm about to give you is in keeping with that philosophy about innovation. And I want to change your minds, because if we're going to change outcomes from surgery, if we're going to prevent complications from surgery, we have to do things differently. And the gut microbiome is an interesting technique or tool or chance for us to be able to do things differently. So very briefly, to explain the language of microbiome science, we used to think that we could not culture the organisms in our gut because they are very fastidious, they require specific culture conditions. We thought maybe we could only culture 30%. This is probably not true. We can probably culture more than this now with advanced techniques, maybe up to 70%. But it was the birth of high-throughput sequencing, and particularly 16S sequencing, that allows us to study the ecology of the gut for the first time in real detail. And this gave birth to the idea of the metataxonome. So this is the structural ecology, not necessarily the function, but who is there. The metagenome came with deeper shotgun sequencing technologies of the gut, which allowed us to study specific gene functions of organisms and allowed us to have newer insights. And there were a couple of big projects that happened in the early 2000s, like the MetaHit project, the Human Gut Microbiome project, which were effectively mapping projects. They tried to tell us who was there. And they used a number of different techniques, which are illustrated here. But there are general steps that typically culminate in some form of animal validation or prospective validation. The major challenge that we have had is, of course, establishing cause and effect. But where I come from, what we are really interested in is studying the function. So I'm particularly interested in studying the metabolome, the metabolism of these organisms, because I believe this is the best way to engineer the microbiome to improve patient outcome. So this is some work done by my colleague, Professor O'Keefe, who works in Pittsburgh and in Stellenbosch in South Africa. So this is to begin to show you the massive variation in the gut microbiome globally. So within the gut, 4 phyla predominate, and this shows you the difference between an American gut and an African gut. It's completely different. The Prevotella phyla predominate versus the Bacteroides in Americans, and that's because, of course, of the different diet and the different environment that the two populations are exposed to. But we can go much deeper. If we go down to even the individual strain level, so this is 3 individual strains, Faecalibacterium prausnitzii, Eubacterium rectale, and Prevotella copperi. Even within a strain level, we see massive variation between countries. So you can see China is really quite a significant outlier. It dominates the principal component analysis here. But also you can see Spain in green. You are really quite different, even within the same strain. So within this very diverse and complex and large ecosystem, we see immense amounts of variation. And in fact, this variation is so significant that none of us are the same. We probably share somewhere between 1 to 10% of the same bacteria at a strain level between individuals. And the human genome is dwarfed by the gut microbiome. It's dwarfed. There are maybe 30 million genes, we think, in the gut microbiome, compared to 23,000 genes that code an entire human being. One of the key messages of my talk that I want to get across is this idea of complexity. The gut is a complex ecosystem with specific niches and time-dependent variations within it. So we are all familiar with this view of the cecum, the lumen, and the rectum. But we have very niche-specific ecosystems that may reside within microscopic niche variations or indeed macroscopic niche variations. What's in the feces is not the same as what's on the mucosa. What's on pathology is not the same as what's off pathology. And we are not just talking about bacteria. This is perhaps the most important thing. We're talking about viruses, yeasts, archaea, and fungi, all of which interact in a dynamic way. And the most important thing is that we don't even know who is there yet. It is almost entirely unexplored. The virome is probably much greater and more important than the microbiome in terms of bacteria. All of bacterial life on the planet Earth is recycled every 48 hours by phages. And we have no idea of the importance of phages really for human health and advancement. So this is some of my data. This is about 154 patients that we've sequenced out of my cohort at Imperial College in London. And what you can see when we map the mucosa of patients having elective oncological surgery is immense variation in the four phyla that predominate. But we don't understand what being at one end of this spectrum really means. What is the significance in terms of operative outcome? What is the significance in terms of your health? We don't know. There is a word commonly used called dysbiosis. You hear it a lot in the microbiome literature, and it means an imbalance, an ecological disturbance. I think it is a meaningless term. It is a non-scientific term. It tells us nothing of function or mechanism, and it tells us nothing that we can action for our patients. What really do we need to, what real use is dysbiosis if we cannot accurately define it? So I think of the gut as a massive chemical bioreactor. It is a chemical superhighway. And within it, we have multiple channels. So in this instance, I'm thinking of cancer, okay? We have lots of established metabolic pathways which we think are procarcinogenic, some which we think are protective. And of course, this is constantly interacting with our immune system. And the chemical or the molecular signaling languages that the microbiome has are very, very complex and very diverse. But they are very much dependent on what you put into the system. Of course, these bacteria can only metabolize what you choose to eat and expose them to. And we can think about this in surgical terms. We can think about a surgical exposome. As soon as we bring the patient into the hospital, we change the metabolites and the fuels that these bacteria are exposed to, and that in turn changes their function. So if we think about things like amino acids which are produced and metabolized by bacteria, change the inflammatory properties of the mucosa, for example. And this is just one example of many different ways. So quite often in surgery, we think about bacteria as translocating, we think about sepsis, we think about antisepsis, but actually their functions are much, much more subtle, much more complicated, and much more diverse than we give them credit for. This is where I'm from in London. This is Imperial College, and every day I walk past this plaque, reminding me that I have a lot of work to do. I want to propose a new system, then, to think about the gut and to think about bacteria. So I think of the gut as a super system. It's a complex super system. And every time you operate on that patient of yours, you perturb that super system in a really complicated way that is really difficult to measure and objectively compare. So as I said, the majority of the perturbations you cause are actually microbial. They're not human. You don't modify the human genome when you operate. Of course you don't. But you expose that patient to a dynamic set of interventions, most of which are non-operative. They're nutritional, they're drug-related, they're contaminant. You might put an implant in a patient. And you do that in a dynamic way. It changes for every patient. And every intervention that you make over time will influence that relationship in a complicated or subtle way, in a network way, or in a systems way, and that will have an output. It will have an effect. One of those effects might be an anastomotic leak. And that's why prehabilitation works. It's why enhanced recovery works. It's why the biggest advances in surgery have been non-technical. So let's just examine briefly some of the major drivers that change the gut microbiome and influence the type of bacteria that your patients will have when you operate. So this is some work that my PhD student, James Alexander, did, and we're very interested in trying to understand how bacteria cause cancer. So we went and sampled about 174 patients. We had a UK cohort and we had a Czech cohort. And what we've done is we've mapped two clusters. This is a benign cluster. These are typically commensal organisms that are friendly versus what we think are kind of slightly nastier bacteria, which we've classed pathobionts, which are typically oral and found in the mouth. And what you find as you go from normal through low-grade, high-grade, stage 1, 2, and 3, 4 cancer is almost a linear drop-off of the benign cluster and a linear rise in these pathobionts. And on a multivariate analysis, the most important determinant of that is cancer stage. Increasingly, we think actually it's the molecular biology of the cancer that defines the ecological niche of bugs that grows within it. But trying to unpick what these bacteria are doing is very difficult. So we took the same patients and we sequenced them to see what genes were being expressed in these cancers. And what we found is a non-linear relationship between which bacteria are associated, and I say associated, with established driver mutations of cancer or oncogenic gene expression. It's not linear. So these bugs are working in a complex network way. This is very controversial. And so we had a meeting in London in 2017, and we brought together these experts to try and discuss whether or not we really think there's evidence to prove at the moment that the gut microbiome drives cancer. And the summary conclusion is we're still not sure. There's not enough evidence. But we just published this in Gut, and it was an awful lot of work, and I must thank Alistair Scott, who did a lot of that writing. But our thoughts are there. So pathology is number one. Number two is diet. Diet is obviously important. So this is a study I did with my colleague Steve O'Keefe and some of the guys in Holland. So we did a trial where we took African Americans from Pittsburgh, who have the highest rates of cancer detection in the United States, versus native Africans. So these were rural Africans from ZuLu-Natal, who have the lowest rates of polyp detection globally. We gave them a colonoscopy, and then we locked them into a house. And we very carefully controlled their diets. And after 2 weeks of a home environment diet, we crossed over their diets. And then we recolonoscoped them, and then we serially sampled them. And what we found was quite astonishing. So within 2 weeks of adopting an African diet, Ki-67 expression dropped in half, CD3 expression, CD68 expression significantly dropped. And in the Africans, it went the other way. So actually, what's important is that these inflammatory biomarkers are modifiable. What we found was that the structure of the microbiome didn't change dramatically, but the network function changed very dramatically. So basically, you went, if you were an American, from a proteolytic network to a saccharolytic network and vice versa. And the metabolic consequences of this are really dramatic. So bile acid excretion in rural Africans went up by 400%. As soon as they started eating massive amounts of protein, which they wouldn't normally adopt. Similarly, short-chain fatty acid metabolism fell off the chart. So we think it's the metabolic network effect of these bacteria which are critical. And every patient that you operate on has drugs. Drugs have a very dramatic effect on the structure and function of the gut microbiome. No greater example than inflammatory bowel disease. So anti-TNF drugs significantly change the diversity and ecology of the gut. And you can see here different taxa of bacteria, bifidobacterium, clostridial bacteria. Blue is responders and pink is non-responders. So in responders and non-responders, the whole microbiome changes. We don't know what the significance of that is. But there are unintended consequences. So as a surgeon, I'm a colorectal surgeon. This is my view of an operating room when I'm doing an open resection. And when you're operating, you're fixed, you're concentrating, you're focused. You're like that picture of Dr. Torre that you saw earlier. You know, you're in the zone and you're focusing. My question is what we're doing in the operating room really the critical factor that's going to define whether our patients leak or don't leak? Of course, technical procedure is critical. You have to have a really precise and good technique. But sometimes, you know, you just do a really great job. You leave the operating room thinking, yes, I smashed that. I did it brilliantly. And sometimes the patient doesn't do well, and you think, why? Maybe it's not your fault. Maybe it's not your fault. We obsess about perfusion. We obsess about blood supply because this is what we are taught. It is the most important thing. And this is from my good friend, John Alverdi, who I'm sure you all know. He dug out this paper from the 1950s. Wonderful experiment, a dog experiment. They completely devascularized some dog colon, as you can see, and they put a catheter into the colon, and in one cohort of dogs, they infused antibiotics, and in the other cohort of dogs, they gave no antibiotics. In a completely ischemic colon, if you don't have antibiotics, everybody dies, and this is what your colon looks like. It falls apart. If you have antibiotics, no dogs died, every anastomosis healed. So it's not just blood supply. It's more complicated than that. Blood supply is important. So John's work has really been in defining the metabolic or molecular functions of pathobionts, of target bacteria that function on the mucosa. And his theory is that actually what happens is that we, through either not great technique or because of poor blood supply, that specific strains of bacteria predominate, and his work is really focused on Enterococcus faecalis, which through an MMP9 mediated process start to metabolize and break down type 4 collagen. So he thinks that literally bacteria are chewing and destroying collagen, and if you've got lots and lots of these bacteria, it doesn't matter what you do, they're going to break down that collagen and that join will fail. And he's very nicely demonstrated that you can transfect that bug into an animal model and you can cause an anastomotic leak every single time if Enterococcus faecalis is there. But the abundances are sometimes 400 times that what we would typically see in a patient. And we have to be very careful of animal models in the human gut. I think it's more subtle than this. I think perfusion and oxygen tension is critically important, but I think the relationship is more complicated. So I think this is a really nice paper which I highly recommend to you, looking at intestinal healing in intestinal injury. So probably what happens is that the microbial organ that we have, these, which are predominantly obligate anaerobes, produce lots and lots of lovely metabolites which maintain the health of the mucosa and of the gut, and they function within a specific oxygen gradient. The problem is that when you disrupt bacteria, through antibiotic use, through surgical disruption, through mechanical bowel prep, you change the way that those bugs metabolize, and that places an anaerobic stress on tissue. And it may be that if your patient is not well perfused, that actually that's compromising the patient in some way. And similarly, when you have excessive epithelial repair, for example, during an anastomotic healing or during wound healing, that in turn triggers microbial organ dysfunction. And so quite quickly you can get into an organ cycle. So this is not, I think John's work is absolutely amazing. It's fantastic. It has energized the surgical community and created very clear focus. But I think it's more complex than that. I think it is more subtle. What are the oncological implications of this? Well, so again, John's group think that maybe we all know that anastomotic leaks do worse. If you have an anastomotic leak, your local recurrence rate goes up, your systemic recurrence rates go up, your outcome is worse. So he's thinking, well, maybe bacteria are important. Well, maybe they are. We don't know yet. But his argument with those same molecular processes that he's identified may allow shed cancer cells to stream out of the gut and drive local recurrence through a macrophage-mediated local immune response. That's a theory. We don't know that yet. But what we do know, if you look at the metastatic disease process, is that metastases are not sterile. So this was a paper published in Science, I think at the beginning of last year. And what this is showing is they looked at a data registry retrospectively in prospective patients. So if you look at your colorectal primary, your Fusobacterium genus is highly expressed, but it's also highly expressed in liver metastases. And so what they then did was they went into an animal model, in a PDX animal model, and they pre-treated these animals with metronidazole. And actually, if you give metronidazole, tumor volumes decrease. So there's a theory at the moment that the gut microbiome drives the metastatic response. And it's an open field now as to what we do with this information and how we work it. So I want to very briefly return to operative outcomes. And I want to think a little bit more broadly. So this guy, who I'm sure you all know, you take his oath, believed that infection happened because bacteria were transmitted through cold air. Well, in fact, he didn't know about bacteria. He thought infection was caused by cold air. And we thought that way for many, many years. And then this guy came along, Joseph Lister, and he developed this concept of antisepsis, and he revolutionized surgery. And then Alexander Fleming created antibiotics in the 1900s, and that transformed outcomes yet again. And we've become completely dependent on antibiotics in colorectal surgery. We all use them. But we've got a massive problem, which is that they don't work anymore. Right? So in the space of 200 years, this amazing wonder treatment that we have has stopped working. And nearly half of all infections caused by pathogens in colorectal surgery are now resistant to first-line antibiotics. If you go through the literature, Fleming knew this. He understood that actually resistance was going to be a problem. And I noticed that he came to see you guys in 1948. I saw this today. I was very excited to see this. So clearly, you guys in Barcelona, you knew about this as well in 1948. So, we are facing Armageddon. And what is the surgical response to this? It's just to keep giving more antibiotics. Okay? We're just going to keep giving more antibiotics. This paper came out. You may have seen this. This was NSQIP data that was published in 2015. It had a huge impact. And it was retrospective data of a large number of patients. And it showed really compelling data that if you give mechanical bowel prep with oral antibiotics, your wound infection rate drops, your ileus rate drops, your anastomotic leak rate drops, and your mortality drops. So the trend in the US and now in the UK, where I'm from, is to give oral selective decontamination. It's to kill everything. This is a bad idea. Okay? So, I'll explain why I think this is a bad idea. This guy is in Azerbaijan. He's sitting in oil. Don't panic. It's oil. Okay? It's oil. So, this is a graph, and it shows you what happens to diversity within the gut when you give mechanical bowel preparation or you give antibiotics. The main thing to notice is that if you give antibiotics, actually diversity in some cases never recovers. You may never return your patients back to normal gut ecology if you give an antibiotic. It tells us nothing about function. It tells us nothing about what the impact is of this for the patient longer term. This is our data on patients who had bowel preparation. The significance of this is that these patients had pre- and post-intervention of analysis of mucosal microbiomes, of the mucosal microbiome. And what we found actually is that if you look at the mucosal microbiome, there's no real change in diversity across populations. It doesn't really change significantly. But what does change is within patient variation. And it's the only thing that really dramatically changes. So if you want to look across populations, it's not helpful. You have to look within individuals. But actually, mechanical bowel prep does have a big effect. This paper came out last year. It's a really good paper. I really recommend it. And what it shows us is that actually the long-lasting effects on the microbiome are quite significant, at least in a mouse model. And laxatives or osmotic agents like polyethylene glycol really do dramatically affect it. They disrupt the mucosa in quite a significant way. They fundamentally influence the immune response. And in fact, actually, they were able to show very nicely that it completely destroys the mucosa, which is the mucus layer that protects the gut lining, and in fact brings bacteria into direct contact with the epithelium. That might be really important if what Dr. Alverdi's work says about the healing mucosa is true. The second point that I would make is that the antibiotic data is poor. This is my map of all recent randomized controlled trials looking at selective decontamination in randomized controlled trials for colonic surgery. Everything in red suggests to me that they haven't properly thought about what they were doing or reported on that data. They haven't thought about it like microbiome scientists. It's not their fault. They thought about it like surgeons. Because as surgeons, we just want our patients to do well. If I give the drug, does my patient do well, yes or no? But they typically have multiple pathologies, and I've told you that pathology is important and defines the microbiome. They haven't really accounted for minimally invasive. They haven't accounted for enhanced recovery. They nearly always never report mechanism. They typically give a varying amount of bowel prep, and they really report variably on SSI leak or SSI or leak. And if you look at the two recent studies from Japan, one has shown it does reduce SSI, one has shown it doesn't. Both have shown no change on leak. I started my talk by telling you that the gut was a complex system. When you perturb a complex system, there will be unintended consequences. We haven't begun to measure those consequences yet. But a really good example is drug metabolism. Bacteria are critical for drug metabolism in ways that we are just beginning to understand. And we define this through what we've called the timer mechanism. And this is ways that the bacteria modify either the efficacy or toxicity of chemotherapy. A good example is irinotecan. E. coli produces a class of enzyme called beta-glucuronidase. It reactivates irinotecan in the gut, and it causes the toxicity and the diarrhea of irinotecan. It's got nothing to do with your genes. It's got nothing to do with your metabolism. It's all to do with bacterial metabolism. The critical importance of that is you can knock it down. You can knock it out. You can prevent diarrhea. The really exciting work that has come out recently has been on monoclonal antibody treatment. And a couple of really big papers came out recently in Science by Laurence Zivogel's group, particularly in PD1, suggesting that the gut microbiome is a critical media of this. She's firmly of the opinion that as surgeons, we have to be very, very careful about how much terminal ileum we remove, because she believes that it's the terminal ileal gut microbiome that determines drug response to things like oxaliplatin and to biological agents that we use in surgery. So we need a new way to prep the gut. We need a new way to think about the gut. How are we going to do it? What's the solution? Well, this is a nice picture of a couple who were living in South America, and over a period of time, they were able to completely change their environment by colonizing it with trees. We can do the same thing in the gut. Instead, what we do at the moment is we just do this, right? This is what we think is what we want for surgery, everything dead. So what happens when you do that is that when the gut recolonizes, you don't necessarily recolonize with lovely green vegetation and a healthy forest and a healthy ecosystem. You predominate potentially pathobacteria like gamma proteobacteria, proteobacterial species, things which perhaps aren't so good for us. And actually, we quite often suppress that over a long period of time with antibiotics, and we give a bunch of drugs that have a further effect, like opiates and things like that. Maybe what we need to do is to actually have a more precise way. We need to maybe be thinking more as ecologists. Can we suppress the bad bugs in our system and actually potentiate the commensal bugs that we need in a slightly more targeted or personalized way? So we need to think differently. To understand the gut microbiome, we need new experimental models. This means longitudinal modeling of individual patients, because I've told you that inter-individual variation is so significant. But also we need new tools for real-time modeling. We can't wait 72 hours for a culture anymore. We need new solutions. And at the moment, no drug trials in cancer, no surgical trials really account for microbiome function in determining the outcomes. So we have to think about this going forward. The good news is that we have lots of new tools at our disposal. We don't just have to rely on antibiotics. Phage therapy is going to be very important. Within our lifetime, that is going to be potentially a solution to the antibiotic resistance crisis. But synthetic biologists and engineers are creating bacteria now that are going to be able to target the microbiome. This is a fundamental shift. You're not giving a drug to change human metabolism or to change human function. You're targeting the bacteria that live there. The microbiome will become a target. It may be that we need to really think about our operative nutritional strategies. Maybe carbohydrate loading is not enough. Maybe we need to be thinking about how we optimize the health of the colonic mucosa. And maybe targeted antibiotics are the way forward. Maybe we should be putting those in extracellular matrices and actually putting them on the site of the anastomoses. What we're interested in our group is trying to understand, trying to develop new microbiome-based biomarkers. And what we're interested in is trying to define healing phenotypes. So I've said that the microbiome is a dynamic change, right? So we think about it in three domains: time, space, and the surgical exposome. It's structured by the time you're 3, and then it changes gradually by the time you're about, over your lifetime. And by the time you're 70, it's very different to the one that you started out with. The problem is the patients that we operate on are perturbed. They're somewhere here, and we don't know where they are. And of course, everyone has a slightly different trajectory, and as soon as you operate, we don't really know where we're starting from, and we go in 3 different directions. We can either be a super healer. There are some people you can spit on the anastomosis and it will heal. There are some people that are probably susceptible, and there are some people that no matter what you do, they're going to leak, right? We all know who they are. We all have those patients. And what we're interested in is trying to define them and trying to understand these different modifiers of the gut microbiome that may in fact define those leaks. So one of the ways that we're doing this is by doing intraoperative or in vivo mass spectrometry, and I will just explain this to you now. Hopefully this video will play. So we are working to take mass spectrometers out of the chemistry lab into the operating room. Mass spectrometry has been used for many, many decades for microbiology. So when you use electrocautery, you create smoke. We take the smoke, we put it into a mass spectrometer. It does all of our sample preparation for us, and it does it in near real time. We have modified our handheld instrument so it has a chimney to suck smoke in. This is, of course, a completely unrepresentative model. I'm sure if there are any liver surgeons in the room, they will be horrified. But effectively, the initial user case was that we could detect cancer margins in real time, and that's pretty much how we've used it. The technology works by aspirating smoke into the mass spectrometer, and then we have a histologically validated database, and then we apply machine learning tools to those data sets in real time to tell the surgeon yes or no, it's cancer, it's not cancer. And we thought, well, maybe we could apply the same thing to studying microbiology. This is data just to show you how it works in an endoscope. So we're funded to develop an endoscopic application. I'm sorry, the video is a bit slow. But you can see a hot snare of an adenoma, and you'll see in a second that we get spectral data that comes out of it. That's what it looks like. Within that data set is lots of bacterial information. It tells us what bacteria are on the mucosa. And this is the same video, but what this shows is the information that the endoscopist will see. So we're able to tell the endoscopist about the nature of the tissue that they're removing. In this case, it's an adenoma, right? So that's kind of real-time chemistry. But what's interesting is that different bacteria, so different classes of bacteria, have very discrete spectra. And actually, we can build models that can tell us in real time which bacteria are present in complex cultures based on near real-time analysis using mass spectrometry. And for us, this is very exciting. We've just been funded by the European Union to develop this, to study the microbiome on anastomoses. We have a PhD student at the moment who's doing prospective analyses. If you're interested and you'd like to come and do your PhD with us, please do tell me, because we are recruiting. So this is some data. We just presented this at DDW the other week. So this is the same technology from a Dutch trial looking at anastomotic leaks. And when we do the same technology and we apply it to feces, we can identify patients who are going to develop an ileus preoperatively based on the metabolic content of their feces. And we think part of the reason that we can do that is because we're measuring the metabolic function of these bacteria within the gut. Interestingly, the ecology in itself was not predictive. And now we have candidate molecules that we're able to target. This is a quantitative chemical technique. It is not a black box technology. So this is the future of our research, real-time phenotyping of the gut during surgery, and we're always looking to collaborate. So I'm sorry for talking a bit too long, but I will summarize my talk. So the microbiome is highly individualized. It is anatomically discrete and niche specific. We are at the very beginning of our journey of understanding what it does, and it's important in human health. But I think it has a very great potential impact in surgery. The most important reason why I think it is important is because it is modifiable. You can engineer it, you can target it, you can change it. And if we can change it for the benefit of our patients, that is a good thing. Lots of people have done this work. Many thanks for your kind invitation. It's a fantastic honor to be here. And of course, we'll take any questions if you have them. Thank you. Thank you. Thank you. Thank you.
Thank you very much, James, for this fantastic talk. I'm sure that we all have enjoyed it a lot. And well, we have understood that when talking about microbiome and surgical outcomes, there are no rapid answers, and the word is complexity. Bueno, si hay alguien que tiene alguna pregunta, y si no, después, a continuación, pues cuando salgamos de la sala también podrán hacérsela. Vamos a continuar entonces con la siguiente parte de este acto, con la segunda parte, que consiste en la entrega del premio al mejor residente del curso académico 2018-2019. Los criterios para la elección de este premio están basados en las diferentes evaluaciones a lo largo del año, de las distintas rotaciones, y también en un examen final que hacemos al acabar el año de residencia para todos los años de residencia. El ganador o la ganadora este año entregará el premio de la doctora Marta Pascual, es la doctora Alba González. Tengo que decir, el premio consiste en un curso de formación que el residente elige. Y ya pasamos a la tercera y última parte de este memorial, que consiste en la entrega del premio Dr. Lino Torre a la investigación quirúrgica. En este caso es el 20º premio Dr. Lino Torre. Y voy a leer directamente el acta de la resolución del 20º Premi Lino Torre, reunidos en Barcelona el 28 de mayo de 2019 a las 9:30 horas en el despacho del director médico asistencial, los miembros del tribunal, el doctor Manuel Pera, el doctor Miguel Pera, el doctor Julio Pascual y también la doctora Pilar Torre, constituidos, manifiestan. Quieren dejar constancia, en primer lugar, de la gran calidad de los trabajos presentados en esta convocatoria y, después de haberlos valorado y calificado de forma individual, resuelven que los criterios para la evaluación, evidentemente, son la calidad de la revista en la que se publican, el diseño del estudio, el impacto clínico y que el primer o último firmante sea un cirujano. Resuelven conceder por unanimidad el 20º Premio Lino Torre al trabajo titulado Comparison of Perioperative Outcomes between Laparoscopic and Open Approach for Pancreatic Duodenectomy, de Padua Randomized Control Trial, publicado en la revista Annals of Surgery en noviembre del 2018, del que es primer firmante el doctor Ignasi Poves, en representación de un equipo formado por miembros de la sección de cirugía hepatobiliopancreática del Servicio de Cirugía General del Hospital del Mar y con la colaboración de miembros de los servicios de anatomía patológica, radiología, digestivo y oncología. Las razones principales de esta decisión se centran en el impacto clínico de un estudio que demuestra que incluso en la cirugía oncológica de alta complejidad es posible obtener beneficios de un abordaje mínimamente invasivo. El diseño en forma de ensayo clínico aleatorizado y la publicación en la mejor revista internacional de cirugía han sido factores determinantes en la elección frente a otros trabajos presentados en la presente convocatoria, todos ellos de una gran calidad. Así que enhorabuena, doctor Poves, doctora Pilar Torres, si puede pasar el premio. Y a continuación, el doctor Poves hará una breve presentación del trabajo.
Buenos días. S'ha de desconnectar... ¿Cómo lo has puesto? Sí, está aquí. Vale, y este... Mejor no lo sé hacer. No.
Ya está bien. A ver,
Está aquí.
te lo voy a... Vale.
I aquest aquí? Aquest l'he ficat aquí, però jo no sé si això va aquí. Ahora, perfecto. Bien. Buenos días, gracias a todos por estar aquí. Antes que nada, pues agradecer al hospital, al servicio de cirugía, toda la colaboración que hemos tenido siempre para hacer todos estos estudios que son bastante complejos, y desde luego es un honor el que después de haber hecho el estudio, de haberlo publicado, pues que encima pueda obtener un premio como este, por lo cual muchas gracias a todos. Este estudio nace hace 10 años. Una cosa es que se empiecen a reclutar pacientes en el año 2013, pero otra cosa es que desde hacía ya una década teníamos en mente que este estudio, que parecía un sueño, hace 10 años, pues se pudiera llevar a cabo, desde luego, con bastantes dificultades. Nuestro objetivo era comparar aquellos pacientes que se operaban de duodenopancreatectomía cefálica, los que no seáis cirujanos. Es una operación conocida como Whipple, que es probablemente una de las más complejas en el mundo de la cirugía, y comparar si entre hacerlo abierto, que es el estándar habitual y sigue siendo el estándar hoy día, hay alguna diferencia, alguna ventaja al hacerlo por vía laparoscópica. La duodenopancreatectomía cefálica, con todos los compañeros, a todo el mundo nos va muy bien, ¿no? A todo el mundo nos va muy bien, pero siempre hay algún paciente ingresado con alguna complicación, y ahora, gracias a la tecnología, a las bases de datos obligatorias, a los registros nacionales, sabemos que la mortalidad en países, la mortalidad de la duodenopancreatectomía en países como Francia es de un 9%, Alemania de un 10%, y en un registro nacional que hicimos nosotros en el año 2015 en España, la mortalidad a 90 días era de un 9%. O sea, estos son nuestros datos actuales. Es decir, estamos ante una intervención quirúrgica con una mortalidad potencial muy elevada. De manera que cualquier esfuerzo por intentar mejorar los resultados, pues sea bienvenido. Nosotros miramos a ver si el abordaje laparoscópico podía tener alguna influencia en mejorar estos resultados. La primera duodenopancreatectomía cefálica se hizo en el año 1992, la hizo un supercirujano, bueno, en aquel momento no era supercirujano porque era el inicio de la laparoscopia. Hoy día es un supercirujano porque ha hecho muchas técnicas quirúrgicas por laparoscopia, pero él dijo que esto no se tenía que hacer por laparoscopia, que era muy complejo y que no aportaba ningún beneficio. Estamos hablando en el año 92, obviamente conocimientos, tecnología, visión ha cambiado mucho y probablemente este cirujano ahora no diría lo mismo. Pero no fue hasta el año 2007 en que se hizo un estudio que impactó enormemente, porque presentó unos resultados más que excelentes de la duodenopancreatectomía por laparoscopia, un grupo indio, un grupo de Palani-Belu, que ha seguido haciendo esta técnica y que ya la tiene estandarizada en su arsenal. En Estados Unidos, que nunca tuvo mucho interés ni seguimiento, hasta que apareció un cirujano, Michael Kendrick, de la Clínica Mayo, es un centro de referencia, en el cual presentó 63 pacientes intervenidos con unos resultados también excelentes. Todo esto, a partir del año 2010, sobre todo la publicación del grupo americano, existe una multiplicación exponencial de gente interesada en realizar esa técnica por vía laparoscópica. Tanto es así que 5 años después, varios autores, haciendo análisis de otros estudios publicados, no con resultados tan buenos, incluso con resultados malos, pues dan en 2 artículos interesantes unos pequeños toques de atención, como diciendo quizá esto no es tan bueno como parece, o quizá no es reproducible como otros cirujanos sí lo puedan hacerlo. Lo que sí es cierto es que con el tiempo los cirujanos han ido cogiendo experiencia, hemos aprendido unos de otros y en estudios prospectivos, de case match análisis, metaanálisis, lo que se ha demostrado es que parece que la laparoscopia al menos no es inferior al abordaje laparoscópico. No obstante, no había ningún estudio prospectivo randomizado publicado ni puesto en marcha tampoco, cosa que es lógica porque ni son muchos los cirujanos que hacen la cirugía del páncreas por laparoscopia, y menos pues hacía 10 años, ni hay tantos casos como por ejemplo cáncer de colon para hacer un estudio, ¿no? El primer estudio que se publicó, el primer estudio que se publicó fue en el BGS, en el año 2017, y fue el mismo grupo indio que en el 2007 había tenido unos resultados tan buenos con la DPC laparoscópica, que hizo el primer estudio prospectivo randomizado. Hay que decir que este estudio, los pacientes están bastante seleccionados en el sentido de que son solo tumores periampulares, no son todas las patologías pancreáticas, y de hecho solo un 15% de los pacientes operados están operados por un cáncer de páncreas, con lo cual pues, bueno, es un estudio un poquito sesgado. Lo demuestra que la edad media de los pacientes, tanto en el grupo abierto como en el laparoscópico, es de 60 años, ¿no? Es un grupo realmente muy joven. Aún así encuentran diferencias en la estancia hospitalaria, que es menor en el grupo laparoscópico que en el grupo abierto, de 13 a 7 días. Y posteriormente se han publicado dos estudios. El nuestro es el PADULAB, que a diferencia del anterior no seleccionamos los pacientes por patología, solo los que cumplan los criterios de selección. Y el estudio LEOPARD 2, que está realizado en Holanda, es un estudio multicéntrico, 4 hospitales, en el que tampoco hacen una selección de los pacientes fuera de los criterios. Y este estudio, les tengo que avisar ya, o les explicaremos que se tuvo que cerrar con antelación por el exceso de mortalidad en el grupo laparoscopia. O sea, ya estamos viendo que probablemente no esté todo solucionado. Bueno, nuestro estudio lo que pretendía era comparar el ADPC, el Whipple, de una manera rutinaria en un estudio prospectivo randomizado. Lo más importante es que se diseñó como intención de tratamiento, es decir, todos aquellos pacientes que se convertían, obviamente no los excluíamos del grupo. Aquellos pacientes, los resultados de la conversión, si el paciente está operado por laparoscopia, están dentro del grupo laparoscopia. O sea, no se han intentado no seleccionar los casos puros de laparoscopia, sino todo el grupo que es el paciente que se intenta realizar por laparoscopia. Todos ellos operados en nuestro centro, y tardamos 4 años y medio en recoger todos estos casos. Criterios de inclusión: paciente que tuviera que realizarse una duodenopancreatectomía cefálica de más de 12 años, de más de 18 años de edad, y obviamente que firmara el consentimiento informado. No sabe cuál será una lesión premaligna, maligna o benigna. Excluimos a aquellos pacientes con enfermedad metastásica o tumores muy avanzados que requerirían, que podrían ser resecables o con neoadyuvancia previa y reducción de la masa tumoral. Estos pacientes no se incluyen en el estudio, o si ya se veía que por necesidad se tendría que hacer una resección vascular con reconstrucción, o aquellos pacientes pues, que obviamente tienen un abdomen claramente hostil para la laparoscopia, como puede ser una eventración muy importante del abdomen, o aquellos pacientes que lo rechazaron. En cuanto al diseño, lo que más tenemos que destacar, y que es uno de los puntos débiles del estudio, es que mientras el abordaje abierto pues, se hacía por cualquier cirujano del equipo, el abordaje laparoscópico se individualizó solo en un cirujano. ¿Por qué? Pues porque la casuística es escasa y incluir un segundo cirujano en la curva de aprendizaje pues probablemente podría influenciar. Aún así, en el 80 o más % de los casos, tanto laparoscopia como abierto, los dos cirujanos se ayudaban entre sí, o sea, que yo lo consideraría una cirugía de grupo. La técnica quirúrgica se hizo la misma en el abordaje laparoscópico que en el abierto, y se tuvo que buscar un endpoint, ¿no?, en el cual se consideró la estancia posoperatoria como el más factible de obtener. Aún así se miraron otros objetivos, que eran el tiempo operatorio, el requerimiento de transfusión sanguínea, las complicaciones posoperatorias, si eran mayores o menores en un grupo o en otro, y sin olvidarnos de la calidad de resección quirúrgica. Como complicaciones, porque esto es importante definirlo, y es uno de los problemas que hay en la mayoría de los trabajos, que esto se está solucionando, había que unificarlas. Entonces, en cirugía general nosotros estamos habituados a evaluar las complicaciones con la clasificación de Clavien-Dindo, que va de 1, complicación más leve, a 5, que sería el fallecimiento del paciente, 1, 2, 3, 4, 5, y a partir de 3 incluido, se considera una complicación severa. Además, existe el CCCI, que es un índice, un score, una puntuación que se obtiene de sumar las distintas complicaciones, porque es raro que un paciente solo haga una complicación. Puede hacer una complicación mayor, una número 4, pero a lo mejor tiene 3 doses y un 1. Y esto también nos ayuda a evaluar si a lo mejor el sumatorio de complicaciones es menor en un grupo o en otro. Y después, un score, bueno, una puntuación que no se utiliza mucho, pero yo creo que es útil, porque hay pacientes que tienen complicaciones graves durante el ingreso y hay pacientes que no las tienen, pero muchos que no los tienen no son graves, pero están ingresados muchos días. Entonces, a estos pacientes también los definimos como mala calidad. Y todo esto, mala calidad de seguimiento. Y todo esto evaluado, y también es importante, a 90 días, no a 30 días, que es como suele ser en muchos estudios. Seguimos una estandarización del análisis de la pieza patológica, en que lo más importante es evaluar los márgenes de resección y el número de ganglios resecados. Todos los pacientes que les toca al grupo laparoscopia, el intento es hacerlo totalmente por laparoscopia. Calculamos 66 pacientes para obtener una reducción del 50% de la estancia hospitalaria, y para esto nos basamos en la estancia previa que teníamos en el centro. En cuanto a los pacientes, y ahora ya esto es lo más interesante, de 86 pacientes tuvimos que excluir 20 y randomizamos 66. De estos 66, de 34 en el grupo laparoscopia, en 2 no se completó la intervención. Normalmente pues son pacientes con metástasis hepáticas, entonces no procede a hacer la resección de la cabeza del páncreas. Y en el grupo abierto no se resecaron 3 pacientes. Obviamente todos los pacientes siguen el seguimiento correcto y de los 34 pacientes, 8 se realizaron, pero tuvieron que convertirse. Una tasa de conversión pues, de casi un 25% alta. Así, finalmente, analizaremos 32 pacientes del grupo de laparoscopia a 29 en el grupo abierto. En cuanto al estudio poblacional, fijaros la población que tenemos nosotros. Nuestra media es alrededor de 70 años. Hemos operado pacientes de hasta 86 años. De hecho, más de un tercio de los pacientes tiene más de 80 años. Tuvimos una diferencia en cuanto a sexo. En el grupo de laparoscopia operamos más hombres, más mujeres, y en el grupo abierto más hombres. No sé un poco a qué atribuir esta diferencia. Y por lo demás, en cuanto a comorbilidad, riesgo ASA, cuántos pacientes llevan drenaje preoperatorio, no encontramos ninguna diferencia. Índice de masa corporal medio es de 24. Esto es un poquito sobre técnica quirúrgica y hallazgos, lo cual nos demuestra que no hay diferencias entre ambos grupos en cuanto a si el páncreas era más consistente, el Wirsung era más fino, el tipo de reconstrucción fue la misma. Lo que sí nos llama la atención es que existen un alargamiento del tiempo operatorio muy significativo en el grupo laparoscopia respecto al abierto. Este alargamiento implica más o menos unas 2 horas de tiempo. En cuanto a la calidad de la resección, tanto en un grupo como en otro, la mayoría de los pacientes, como suele ser habitual en todas las series, fueron por patología maligna, siendo el cáncer de páncreas el diagnóstico principal. No encontramos diferencias en cuanto al tamaño tumoral, significativas, y no encontramos diferencias en cuanto al número de ganglios resecados, aunque aquí, en el grupo de laparoscopia se resecaron una media de 15 ganglios, en el grupo abierto 21. Las diferencias no fueron significativas. Hay que decir que en la calidad de resección de la cabeza del páncreas se considera óptimo a partir de 14. O sea, muchas veces no se ha demostrado que resecar 15 o 21 tenga impacto en la mortalidad. En cuanto a la afectación de márgenes, nosotros tenemos un servicio de anatomía patológica que es súper estricto en la evaluación de los márgenes, y si el paciente está, lo que diríamos, limpio de tumor, los márgenes reales son negativos, pero la distancia es de menos de 1 milímetro, se considera una cirugía R1. Por eso, nuestras cifras de cirugía R0, que eso quiere decir radicalidad completa obtenida, son de un 60% y un 50% en algunos grupos. Pero esto no quiere decir que quede tumor. En cuanto a la evaluación de las complicaciones, la más importante en la cirugía del páncreas siempre es la fístula biliar. Es la que, por muy bien que hagas la anastomosis, por muy contento que te hayas quedado, muchas veces después no va bien. No sé si es por el microbioma, pero se está estudiando también todos estos aspectos. Fijaros, en el grupo abierto, un 27% de fístula pancreática, en el grupo de laparoscopia un 12%. Y esto mismo, en el resto de las complicaciones específicas, fístula biliar, vaciamiento gástrico lento, también se ha encontrado que es así, aunque las diferencias no sean significativas. Debería ser una muestra muchísimo más amplia para que salieran. Y en cuanto a la evaluación de complicaciones graves, en general, porque la clasificación de Clavien-Dindo no solo evalúa complicaciones específicas de la técnica ni quirúrgicas, sino, por ejemplo, una neumonía, un infarto, quedan todas aquí registrados. Existen más complicaciones graves en el grupo abierto. Esta sería, al final, lo que serían las tablas importantes y resumen del trabajo, en el cual vemos que en el grupo de laparoscopia existe significativamente una menor tasa de complicaciones graves, Clavien mayor o igual de 3. El score del CCI, que es la tasa acumulativa de Clavien, es menor en el grupo de laparoscopia que en el abierto, lo mismo que en la calidad del posoperatorio, ¿no? 7 pacientes de 32 fueron remitidos en el grupo laparoscopia y 4 de 29 en el grupo abierto. Hay que decir que la tasa de readmisiones en centros tan reconocidos como el Memorial Sloan Kettering de Nueva York es del 25%. O sea, esto que puede parecer enorme, bueno, comparado con otros grupos, no lo es tanto. Solo se tuvo que reoperar un paciente del grupo de laparoscopia y 5 del abierto. No hubo mortalidad en el grupo de laparoscopia y 2 pacientes fallecieron en el grupo laparoscopia. Aún así veis que las diferencias no son significativas porque no está diseñado el estudio para evaluar estas diferencias. Para lo sí que está diseñado es para evaluar la reducción de la estancia hospitalaria, prácticamente en 3 días y medio, ¿no? 3 días y medio en el grupo laparoscopia, y creamos una variable nueva que era aquellos días de estancia más los días que había reingresado el paciente en caso que hubiera reingresado. Esto nos salía una estancia extendida posoperatoria de 14 en el grupo laparoscopia y 18 en el grupo abierto. Como conclusiones, el abordaje laparoscópico tiene dos principales inconvenientes. Primero, un tiempo operatorio largo, de 2 horas. Y 2 horas de laparoscopia es mucho. Más cuando estamos hablando de pasar una cirugía de 6 horas a 8 horas. Esto, obviamente, con la experiencia pues, se puede limitar algo, pero solo algo. La tasa de conversión es de un 23%. Y esto después de haber superado la curva de aprendizaje. O sea, es una tasa de conversión que no es nada baja. Desde luego, seleccionando pacientes, podríamos disminuirla muchísimo. A cambio, existe una igual calidad de la resección oncológica, quirúrgica, una reducción significativa de la estancia hospitalaria de 3,5 días y, sobre todo, una reducción de casi el 50% de las complicaciones graves posoperatorias. Por eso, si algún centro tiene la suficiente experiencia con su equipo quirúrgico de hacer el abordaje del Whipple por laparoscopia, está totalmente aceptado hoy día y pues, se puede considerar incluso el gold estándar. Muchas gracias.
Muchas gracias al doctor Poves por esta presentación tan clara de un excelente trabajo que desde su publicación ha ido ya ganando varios premios, y enhorabuena a todo el equipo. Hemos llegado ya al final de este acto. Simplemente agradecer nuevamente al doctor James Kinross que haya aceptado ser nuestro invitado hoy por una excelente presentación. Felicitar nuevamente a la doctora Alba González y al doctor Poves y todo su equipo por los premios que han recibido y darles las gracias a todos ustedes por su presencia aquí y emplazarles para que nos acompañen nuevamente el próximo año. Muchas gracias.